Sterilization and disinfection. Microorganisms are ubiquitous in nature. Many of them can cause diseases in human and animals. They are also responsible for spoilage of food and other organic materials. Therefore, it is beneficial to kill or inhibit their growth to minimize their harmful effects. This can be achieved by two common methods, that are sterilization and disinfection. Sterilization is defined as a process whereby all viable microorganisms, including bacterial spores, are killed or are completely eliminated. Disinfection involves destruction of pathogenic microorganisms but not their spores on inanimate objects. Sterilization or disinfection can be achieved by means of physical and chemical methods. Physical methods include heat, radiation, and filtration. Sterilization or disinfection achieved by means of chemicals is either in liquid form or gaseous form. Different methods under each category will be discussed in the coming slides, so keep watching. Various objectives of sterilization and disinfection are to prevent microbial contamination of various inanimate objects like instruments, glassware, needle, syringe, culture media, etc. To maintain a sterile environment while performing all microbiological techniques. To ensure sterility and safety in vaccines, drug and food manufacture. And for maintaining asepsis in surgery and medicine. Factors that affect the efficacy of both disinfection and sterilization are prior cleaning of the object, which will help in decreasing the microbial load and removing organic and inorganic matter from an object that needs to be sterilized. It will also render objects safe to handle, use and discard. Number 2 is presence of organic and inorganic load, as they may affect the effectiveness of the process. Number 3 is type and level of microbial contamination. For example, bacterial spores and some viruses like prions require higher temperature to get killed than their vegetative forms. Number 4 is exposure time and concentration of sterilant or disinfectant. And number 5, presence of biofilms that affects the effectiveness of the process by hindering the exposure of contaminated surfaces to sterilant or disinfectant or heat. Let's start with the physical method of sterilization that are achieved through heat. Heat is employed either as dry heat or moist heat. In the coming slides, you will get to learn about each method employing heat as a means of sterilization. First category is dry heat sterilization. It is the process of removing microorganisms by applying moisture-free heat. It is most important, reliable and widely used method of sterilization of heat-resistant articles. Various methods employed to achieve dry heat sterilization includes red heat, flaming, incineration, and hot air oven. Red heat. Red heat sterilization is a process of instant sterilization by holding the instruments in a Bunsen flame till they become red hot. This method ensures effective sterilization. It is commonly used for sterilization of instruments like metal loops, wires, and points of forceps, but is only limited to substances that can endure heating until red in flame. Flaming. Flaming involves exposure of metallic objects to flame for some time, where the flame burns the microbe present on the instrument. This method is used for sterilizing scalpels, needles, mouth of culture tubes, slides, and cover slips. But there are few limitations, like this process doesn't ensure sterility, and the gas flames may leave residues like carbon on the object if it is not heated enough. Incineration. Incineration is the process of sterilization along with a significant reduction in the volume of the wastes. It is an excellent method of removing biohazardous waste. The scraps are heated till they become ash, which are then disposed of safely. This process requires a special device called incinerator, but its excessive use can lead to air pollution. Hot air oven. Standard setting for a hot air oven is 1 hour at 160 degrees Celsius. At this temperature, destruction of all living cells due to oxidation of the cell contents takes place. This is the best method of sterilizing dry glassware such as test tubes, petri dishes, flasks, pipettes, and instruments such as forceps, scalpels, and scissors. It can be used to sterilize liquid, paraffin, powder, fats, and other heat-stable items that are adversely affected by steam. It does not cause rusting of the metal objects, but uniform heating depends upon proper loading of the oven, and further rise in standard temperature may char the paper or cotton plug. The oven is usually heated up by electricity. It has a thermostat that maintains the chamber air constantly at a chosen temperature, a fan or blower to assist the circulation of air to ensure rapid uniform heating of the load. Spores of non-toxigenic strain of Clostridium tetani are used to test the efficiency of dry heat sterilization. Next category is moist heat sterilization. Culture media, aqueous solutions, clothes, rubbers, and other materials that would be damaged by dry heating are sterilized by moist heating. Moist heat kills microorganisms by coagulating of their proteins. There are three main categories of temperature range by which the moist heat sterilization can be achieved. They are temperature below 100 degrees Celsius, temperature at 100 degrees Celsius, and temperature above 100 degrees Celsius. Moist heat sterilization attained at the temperature below 100 degrees Celsius include number 1, pasteurization. In this process, all non-spore forming microbes in the fluids, mainly milk, are killed to enhance the shelf life. Fluids are heated to the temperature of 63 degrees Celsius for 30 minutes, that's also known as Holder's method, and 73 degrees Celsius for 20 seconds, also known as the Flesch method. The process works on the principle of logarithmic reduction in the number of viable microbes so that they can no longer cause diseases. Besides milk, vaccines of non-sporing bacteria are also pasteurized at 60 degrees Celsius for 1 hour in a special water baths. Similarly, serum and body fluids with coagulable proteins are also sterilized at 56 degrees Celsius for 1 hour in water bath. Moist heat sterilization attained at the temperature of 100 degrees Celsius. Number 1 is boiling. It involves boiling of the object at 100 degrees Celsius for 10 to 15 minutes in water. Distilled water is preferred because the hard water might result in the formation of a film of calcium salts on the instruments. The process is simple and useful for removal of most of the pathogenic vegetative microbes and some spores in case no other better method is available. Hence, doesn't ensure complete sterility. Number 2 is tyndallization. Tyndallization is the process of sterilization discovered by John Tyndall to kill the bacterial spores. It involves exposure of materials to the steam at 100 degrees Celsius for 30 minutes for 3 consecutive days. The process is also called as fractional sterilization or intermittent boiling and is accompanied in Arnold steam sterilizer. First exposure to the high temperature kills all the vegetative forms of the microbes, and in the intervals between heating, the remaining spores germinate into vegetative forms, which are killed on subsequent heating. This method is commonly used for sterilization of media with sugar and gelatin, so as to preserve sugar which might get decomposed at higher temperatures. Moist heat sterilization attained at a temperature above 100 degrees Celsius, which includes steam under pressure. Water usually boils at 100 degrees Celsius under normal atmospheric pressure. However, the boiling point of water increases if the pressure is increased. As a result, the steam generated under pressure has a higher penetrating power. When this steam comes in contact with the surface, it kills the microbes by giving off its latent heat during the process of condensation. This principle is employed in an autoclave, where the water boils at 121 degrees Celsius at 15 pounds pressure for 15 minutes. The steam under pressure is used for the sterilization of contaminated instruments, different culture media, and numerous other items that can withstand high temperatures and pressures. The process ensures complete sterility. But heat level substances like oil, powder, and related products cannot be sterilized by moist heat because moisture cannot penetrate into deeper parts of oily materials and powders are destroyed by moisture. To check the efficiency of moist heat sterilization, the indicator commonly used is the paper strips containing spores of Bacillus stereothermophilus. Factors affecting sterilization by means of heat includes nature of heat, temperature and time of application, initial number of microorganisms, nature of microorganisms, type of material to be sterilized, presence of organic material on materials that need to be sterilized. Next physical method of sterilization is by means of radiation. Irradiation is the process of exposing surfaces and objects to different kinds of radiation for the purpose of sterilization. Radiation can kill microorganisms are of two types, namely ionizing and non-ionizing. Radiations are commonly used for sterilizing heat-sensitive materials such as disposable plastic products and materials that cannot withstand heat. First category is the non-ionizing radiation, which includes infrared radiation. These are low energy electromagnetic rays with wavelength longer than those of visible light. It kills microorganisms by oxidation of molecules as a result of heat generation. Infrared radiation is used for rapid mass sterilization of pre-packed items such as syringes and catheters. Second is ultraviolet radiation. UV light has a very little ability to penetrate matter. Thus, only the microorganisms that are exposed directly to it are susceptible to destruction. Hence, it is ineffective in shaded areas including areas under dirt. It is used to sterilize operation theaters, laboratories, entryways, and interiors of biological safety cabinet between uses. Upon exposure, these radiations are absorbed by nucleic acids and cause the formation of pyrimidine dimers which bring error in DNA replication. It can damage some plastics such as polystyrene foam if exposed for a prolonged period of time. It can also damage cornea and skin. Hence, its use in medicine is limited. Ionizing radiation. Ionizing radiations are excellent sterilizing agent. These radiations cause mutations in DNA and produce peroxides. They penetrate deep into the objects. These radiations do not produce heat on the surface of the materials. Hence, sterilization using ionizing radiations is referred to as cold sterilization. It can destroy bacterial endospores and vegetative cells, both prokaryotic and eukaryotic. However, ionizing radiation is not always effective against viruses. Second category of radiation is ionization radiation. It includes gamma rays. These are very penetrating and are commonly used for sterilization of disposable medical equipment such as syringes, needles, cannulas, and IV sets. Gamma radiations require bulk shielding for the safety of the operators. They also require storage of a radioisotope, usually cobalt-60, which continuously emit gamma rays. It cannot be turned off and therefore always presents a hazard in the area of the facility. X-rays. High energy X-rays are a form of ionizing energy that allows sterilizing large packages and medical devices. X-ray sterilization is an electricity-based process not requiring chemical nor radioactive material. High energy and high power X-rays are generated by the X-ray machine that can be turned off for servicing and when not in use. Next physical method of sterilization is by means of filtration. The process of filtration is unique among sterilization techniques in that it removes rather than destroys microorganisms. Thus, further, it is capable of preventing the passage of both viable and non-viable particles and thus can be used for both the clarification and sterilization of liquid and gases. Filtration is an effective and reasonably economical method of sterilization. It is used to sterilize heat-sensitive fluids and air. The primary mechanisms involved in filtration are sieving, adsorption, and trapping within the matrix of the filter. It is particularly useful for sterilizing solutions containing toxins, enzymes, drug, serum, and sugar, as well as air and other gases for supply to aseptic areas. Sugar solutions tend to caramelize during autoclaving and so they are best sterilized by filtration. There are two types of filters, namely membrane filters and depth filters. Membrane filters. Membrane filter is used for filtering liquid culture media or other heat labile liquids. Membrane filters have tiny pores that let the liquid pass through them but prevent the bigger particles such as bacteria from passing through it. The holes and molecules passing through them are precisely controlled. Membrane filters are made up of cellulose acetate, cellulose nitrate, polycarbonate, polyvinylidene fluoride, or other synthetic porous materials. Membrane with a pore size of 0.2 micrometer in diameter are used to remove most of the vegetative microbes, but not viruses. Membrane filters has also an essential role in sterility testing. The filter is first employed to trap and concentrate contaminating organisms from the solution and the test. It is then placed in a liquid nutrient medium and incubated to encourage the growth and turbidity. Second is depth filters. Depth filters are the oldest type of filters. They consist of overlapping layers of fibrous sheets of paper or asbestos or glass fibers. The overlapping fibers create random paths through the filter that trap many particles. That filters are made up of diatomaceous earth in case of bulk-filled filters, unglazed porcelain in case of chambered and filters, and asbestos in case of C-filter. Depth filters, including HEPA filters, are used to remove microorganisms and other contaminants from liquid and air respectively. The air is freed from infection by passing it through high efficiency particle arrestor filter, also known as HEPA filter. Laminar airflow biological safety cabinets are one of the most important air filtration systems. It employs HEPA filters, which can remove 99.97% of 0.33 micrometer particle size. Chemical methods of sterilization and disinfection. It is the process of removal of microorganisms by the use of chemical agents. These methods are used for heat-sensitive materials like plastics, fiber optics, and biological specimens. Chemicals either in liquid or gaseous states are employed. However, it is crucial to ensure that the material undergoing sterilization are compatible with the chemical being used. It is also important to adopt safety rules in the workplace during the use of such chemical agents. Chemical methods of sterilization or disinfection can be categorized as liquid and gases. Various gases and liquids that are used will be discussed further. Ideal properties of the chemical agents. It should have a wide spectrum of activity, should be active in presence of organic matter, should have high penetration power, it should be stable, effective, safe and easy to use, should be cheap and easily available, should not corrode metals. Starting with gases, gaseous sterilization involves the process of exposing equipments or devices to different gases in a closed, heated or pressurized chamber. Gases can be used along with heat treatment. The mechanism of their action is different for different types of gases. Gases include ethylene oxide. Ethylene oxide gas is used for chemical treatment to sterilize or disinfect different types of equipments and surfaces. It has wide range of compatibility with different materials. The mechanism of antimicrobial action of this gas is alkylation of sulfhydryl amino hydroxyl and carboxyl group on proteins and amino group in nucleic acids. Good penetrating nature of gas allows its diffusion into many packaging materials, including rubber, plastics, fabrics, and paper. Ethylene oxide can kill all known viruses, bacteria, including spores, as well as fungi. It is compatible with almost all materials, but is highly flammable, toxic, and carcinogenic in nature. Second is formaldehyde. It is another important highly reactive gas which is used for sterilization and disinfection. It works on same principle as that of ethylene oxide. Formaldehyde gas is obtained by heating formalin 37% weight by volume to the temperature of 70 to 80 degrees Celsius. It is highly microbicidal and can also kill spores. It is used for sterilization of reusable surgical instruments, papers, and cotton fabrics, etc. Formalin is extensively used for the preservation of specimens, for inactivating viruses and bacteria in some vaccines, for example, in original Salk polio vaccine. Formaldehyde is volatile and toxic to both skin contact and inhalation. Next is nitrogen dioxide. Nitrogen dioxide is a rapid and effective sterilant that can be used for the removal of common bacteria, fungi, and even spores. It is effective at standard temperature and pressure. The biocidal action of this gas involves the degradation of DNA by the nitration of phosphate backbone, which results in the lethal effects on the exposed organism. Ozone. Ozone is a highly reactive industrial gas commonly used to sterilize air and water and as a disinfectant for surfaces. Ozone has a potent oxidizing property that is capable of destroying a wide range of organisms, including prions. It is usually generated from medical grade oxygen. Because ozone is an unstable and reactive gas, it has to be produced on site, which limits its use in different settings. It is also very hazardous. Coming to the liquids, liquid sterilization or disinfection is the process which involves the submerging of equipment in the liquid agent to kill all viable microorganisms and their spores. Liquids are not as effective as gases. These are appropriate in conditions where the low level of contamination is present. Number 1 is hydrogen peroxide. Hydrogen peroxide is a strong oxidant and can destroy a wide range of microorganisms. It is useful for the sterilization of temperature-sensitive equipments like endoscopes. Vaporized hydrogen peroxide is used to sterilize largely enclosed and sealed areas, such as entire rooms. But it has a low material compatibility and lower capacity to penetrate. Contact of the liquid solution with skin can cause bleaching or ulceration. Even short-term exposure of the vapor can be hazardous to eyes and respiratory system. Glutaraldehyde. It inactivates microbial proteins by cross-linking the functional groups in the proteins. 2% solution of glutaraldehyde is known as Sidex, which acts as a bactericidal and virucidal in 10 minutes and sporicidal in 3 to 10 hours. Due to low penetrability, the use of glutaraldehyde is limited to surfaces with less contamination. It is used to sterilize hospital equipments. It is volatile and toxic by both skin contact and inhalation. Halogens. Among the halogen, antiseptics and disinfectants are chlorine and iodine. Halogens combine with microbial proteins and inhibit their function. Iodine. Iodine is used as tincture of iodine, which is an alcohol solution. Combination of iodine and organic molecules are called iodophores, known as betadine, which contains a surface active agent called povidone. Iodine compounds are effective against gram-positive bacteria. Chlorine. Chlorine is used as sodium hypochlorite, also known as bleach. It is used to disinfect equipments in dairies, slaughterhouses, and restaurants. The chloramine contains chlorine together with ammonia used to sanitize glassware and eating utensils. Chlorine is also used as a gas to maintain a low microbial count in drinking water. Phenol. One of the first chemicals used for disinfection was phenol by Joseph Lister in the year 1860. It acts as a standard for other disinfectants. Phenol derivatives called phenolics are useful as antiseptics and disinfectants. They damage cell membranes and inactivate enzymes of microorganisms while denaturing their proteins. They include glycols such as lysol as well as several bisphenols such as hexachlorophene. A chemical agent resembling the phenol is chlorhexidine, which is used as skin disinfection as an alternative to hexachlorophene. Alcohols. Alcohols work on the principle based on denaturing or coagulating proteins and dissolving membrane lipids of microorganisms. The type of alcohol most widely used is 70% ethyl alcohol, 70 to 80% ethanol, 60% propanol, and 70% isopropanol. Alcohols are effective against fungi, vegetative bacteria, mycobacterium species, and some lipid-containing viruses, but are not effective against bacterial spores. Isopropyl alcohol, also known as rubbing alcohol, is useful as both antiseptic and disinfectant. Because alcohol evaporates quickly, they leave no residue and are useful in decharming the skin before injections. Heavy metals. Number of heavy metals have antimicrobial property. Heavy metals combine with sulfitryl groups on cellular proteins. Some of them are silver, which is used as silver nitrate in the eyes of newborns to guard against infection by Neisseria gonorrhea. It is also used to cauterize wounds. Next is copper. Copper is used as copper sulfate to retard the growth of algae in swimming pools, fish tanks, and reservoirs. And zinc. Zinc is useful as zinc chloride in mouthwashes and as zinc oxide as an antifungal agent. Soaps and detergents. Soap and detergents are mainly used as a surfactant. They denature proteins, interfere with glycolysis, and damage the membrane of the microorganisms. Depending on the alkali content in the soap, their mode of action differs. Cationic detergents are quaternary ammonium compounds. They solubilize the cell membrane of microorganisms. Among the popular compounds are zephyrin, that is benzalkonium chloride, and cipacol, that is cetylpyridinium chloride. Soap and detergents are effective against vegetative bacteria but are ineffective against spores. This is all about the sterilization and disinfectants. Thank you for watching, and don't forget to subscribe and hit the notification bell to remain updated. Thank you.